Hello, everyone, and thank you for joining our workshop today. My name is Cora Lewis, and I'm a product manager covering routine diagnostics at STEMCELL Technologies. In collaboration with Abbott Molecular, it is our great pleasure to introduce our speakers today, Drs. Murti Vandavali and Farzad Narai. Recurrent chromosomal abnormalities play a central role in B-cell malignancies, and one of the challenges for diagnostic labs can be the detection of these aberrations during low-level or minimal residual disease, MRD. Our first speaker, Dr. Vandavali from the Institute for Cancer Genetics at Columbia University, will be speaking about the utility of CD19 positive cell enrichment upstream of FISH for the detection of cytogenetic abnormalities during B-cell leukemia and lymphoma. Our second speaker, Dr. Farzad Narai from NeoGenomics Laboratories, will be giving an overview of the cytogenetics in multiple myeloma, the technology associated with diagnosing it, and the impact of CD138 positive cell enrichment on chromosomal analysis by FISH. I will now hand things over to our speakers to begin the presentation.
This is Murti Vandavali from Columbia University Medical Center in New York. In this talk, I will present our experience of CD19 enrichment in B-cell malignancies and its use in doing the FISH and its incorporation into cytogenetics workflow. I will also briefly talk about its utility in detecting minimal residual disease, MRD, and detection of subclonal cytogenetic abnormalities. The basis for tumor cell enrichment is the following. Historically, this approach has been used for a long time to enrich tumor cell populations using antibodies specific to surface antigens. This strategy has a paramount relevance in cancer diagnosis, identification of biomarkers for stratification, and monitoring response to treatment. Tumor cell enrichment has huge potential in cancer when combined with genomic applications. Such an advantage has been well established in case of CD138 selection in plasma cell disorders, and this method has become a normal practice in cytogenetics laboratories across the world in identifying recurrent chromosome abnormalities. Although such recurrent abnormalities are also well recognized in B-cell malignancies, tumor cell enrichment for identifying genetic abnormalities in B-cell malignancies is lacking. Particularly, detection of low-level disease is challenging in B-cell malignancies. We have tested CD19 enrichment methods on a large cohort of post-therapy B-cell malignancies to detect known specific chromosome abnormalities. The basis for CD19 selection is that it is a 95 kilodalton transmembrane glycoprotein specific to normal and neoplastic B lymphocytes. It is ubiquitously expressed in neoplastic B cells, used in the diagnosis of B-cell malignancies mainly by flow cytometry. Its use in identifying cytogenetic abnormalities in B-cell malignancies is lacking. The methodologies which are used and the criteria applied in selecting the cases is the following. We have consecutively selected samples in a period of 2 years from pediatric and adult patients with a previously established diagnosis and a B-cell malignancy, and also a known FISH detectable cytogenetic abnormality in these cases. Specimens simultaneously evaluated for CD19 FISH, routine flow cytometry or minimal residual disease MRD, and in some cases with FISH on unselected cells. CD19 selection was performed using STEMCELL Technologies CD19 positive selection kit as per manufacturer's protocols. Eluted cells from the column were subjected to hypotonic treatment and fixed in methanol acetic acid fixative. Protein interface FISH was performed using probes specific to various B-cell malignancies. The following slide shows the list of FISH probes we have used in various B-cell malignancies. The diagnosis, probes used, and their normal cutoffs are shown in this slide. I will not be able to go through in more details, but you can see those in this slide. This cohort consists of 187 specimens from 114 patients. Majority of the samples were from B-cell acute lymphoblastic leukemia and chronic lymphocytic leukemia. A small subset was from the mantle cell lymphoma, marginal zone lymphoma, follicular lymphoma, and other miscellaneous B-cell malignancies. Hereafter, the small group is called as. So how can we improve detection rate of cytogenetic abnormalities using the CD19 enriched cells? So to recognize this, we have first compared the fish on unselected cells versus CD19 selected cells. These plots show the comparison between these two groups in detecting the fish abnormalities. These cases consist of 15 CLL, 9 cases of B acute lymphoblastic leukemia, and 4 others B NHL. This is a subgroup of the samples which we have used in the selection. The differences in the frequency of detection, which is shown here, unselected in the first and then the CD19 in the next column. You can see these differences are highly significant in all the histologic groups. So this validates the benefit of CD19 selection, notably in the cases where the flow frequency, the unselected cells showed low frequency of chromosome abnormalities. This slide shows the similar comparisons between flow and CD19 fish. So CLL cases are shown on two left panels, 13q in the deletion in the first panel and all other probes in the second panel. And the third panel shows BALL and the fourth panel other BNHL. A notable feature here is the most samples with flow positive are positive for CD19 fish identification, but with a higher rate of detection. However, you can notice here there are some samples with flow positivity. For example, this case, these cases are flow positive but negative for CD19, or low detection rate by CD19 fish in number of cases. Vice versa, in some cases flow was negative but CD19 fish could identify positivity. These discrepancies could be technical, artifact, or has biological basis. I will discuss these discrepancies in one of the following slides. So these two methods, despite some discrepancies, complement each other in detecting the disease. So in this plot, the same cases, but selected cases of CD19 fish in samples where the flow identified very low frequency of abnormal cells, shown separately for each category of CLL, BLL, and other BNHN panel, but all cases are combined in the right fourth panel. So here clearly shows all the cases with low frequency of flow positive shows high frequency of fish identification on the CD19 cases. So this comprehensively establishes that CD19 selection has an advantage in detecting the low frequency abnormalities by flow in a significant manner. So this is actually the same plots which I have shown, but the fourth plot with all the cases shown in a log scale frequency. If you look at this here, you can see the low positivity of around 1% or lower than 1% flow positivity shows very high frequency of detection of CD19 uh, FISH positivity. So despite the fact that the promise of CD19 FISH is clear, several key discrepancies between various modalities were seen in this analysis. So I want to address these similarities and discrepancies in this slide. So to address these differences, we analyzed the data as negatives in both comparisons same and positives in both comparisons and discrepant in these comparisons. The blue color shows the negatives and green shows positives. Red shows discrepancy between two comparisons. If you take CLL cases, most of them shows both comparisons in the unselected samples versus CD19, either they're negative or they're positive. But in two cases, you can see that there are discrepancies. Similarly, in flow versus CD19, you see 6 cases discrepancies. Similarly, you will see here few cases show discrepancies. What is the basis for these disagreements? That is what I'm going to show you in this slide. So this slide shows each of these discrepant cases. The green highlighted cases are the ones which is CD19 selection was positive, whereas the flow was negative. So here, if you look at in this, 2 CLL cases and 3 BLL cases where unselected cells showed negative by FISH, whereas CD19 selected cells showed detectable frequency, in some cases high frequency in CD19 FISH. So this is obvious owing to the advantage of CD19 selection. Similarly, between flow and CD19, there are 2 cases in ALL and 1 case in other B NHL showed flow negative, whereas the CD19 cell positives. So this is again the advantage of selection of CD19. So thirdly, there are several cases showed FISH failure in detecting the abnormality, whereas flow was positive. Of these cases, if you look at these 5 cases, these are BCR positive BLL, flow showed low positivity, whereas FISH on CD19 selected cells is a failure. So this points to the challenge that CD19 FISH in this category is not useful in very low frequency of flow positivity or MRD positivity. Thus, this warrants use of other methods such as RT-PCR in detecting the BCR-able positivity. Similarly, in 2 other BNHL cases, you can see there is a flow positivity but FISH negative. These are both cases are IgH cyclin D1 positive cases which fail to detect the abnormalities. So again, the low positivity is a problem in identifying by the CD19 in some categories. In some others, in CLL, there are 4 cases, there are 6 cases showed flow positivity but fail to recognize specific abnormalities in these cases. These, particularly the probes which are used to interrogate the cells, are the ATM and 13q deletions. These abnormalities are known as a secondary abnormalities in CLL. The reason for this failure is most likely that these were present as subclones in the diagnostic specimen, and those subclones might have disappeared in these cases. So that is the reason for the failure. Similarly, in the case of other BNHL, you can see that BCL6 is failed to recognize the abnormality. This is also most likely a secondary abnormality in this case. So this is an obvious pitfall and limit this approach in identifying the genetic abnormalities. So the other thing which I want to talk about, the genomic heterogeneity or clonal heterogeneity. This is a known important fact in cancer biology. Every cancer is unique. They arise clonally and evolve continuously. So this tumor heterogeneity is seen in between the tumors and within the tumor. This clonal heterogeneity shapes the evolution and progression of tumors, affect the key pathways and phenotypes. Inter-tumor heterogeneity poses diagnostic and therapeutic problems, and also causes drug resistance in the tumors which carry the genomic heterogeneity. So its identification is critical in cancers. In the present analysis, we have identified the clonal heterogeneity in 11 out of 76 informative cases. The subclones which were identified in these 7 cases, in these 11 cases, are the following. They are shown individually, and the subclone which is seen are indicated by red markings. So if you look at case number 1, the stem line has trisomy 12 in 60.5% cells, whereas deletion of ATM P53 in 8.5% cells. Similarly, in case number 2, IKZF1 is in lower frequency. In case number 3, ATM P53 deletion 8.5%, and in case number 4, ATM deletion in lower frequency compared to deletion 13q in the stem line. Similarly, in case number 5, ATM deletion as a secondary abnormality and a subclone, and in case number 6 and 7 and 8, 6q deletion is as a subclone. In case number 9, 6q deletion was in stem line, whereas IGH rearrangement was seen in 8% cells as a subclone. In case number 10 and 11, there are 2 different subclones are identified. Those are, in case number 10, 7q deletion monosomy 7 are seen in lower frequency than the BCR-able positive case. In case number 11, ATM was in the stem line, whereas ATM P53 deletion and MYC rearrangement is seen in subclones. So consequently, CD19 fish can provide presumptive evidence for emergence of new subclones, and this is a very important observation. As such, this identification of clonal heterogeneity plays a major role in clinical decisions in the treatment of these patients. So the other observation we made in this limited study of cases is the identification of a minimal residual disease in identifying the early relapse. So the advantage of CD19 fish is in predicting the early response is shown in this slide. Illustrated in this slide is one case of BCR-ABL positive BALL. We followed up this patient for 11 consecutive time points during the treatment. This patient went through remission relapse cycles during the treatment. CD19 fish in these samples are shown in green bars, showed the high detection rate in case where you have flow shown in red color is low frequency. If you can see in 3 time points, which is shown in these yellow stars, this we can see clearly. The low positivity by flow and high rate of abnormality in CD19 selected cells. If you see these comparisons, you can clearly see these significant differences in the flow versus the CD19 identification of the abnormality. So if you see these 3 time points, you can see that immediately after that there is a complete relapse. In this, you can see that in this next follow-up, complete relapse. Similarly, in this, you can see complete relapse. Whereas in 2 other time points, none of the methods have identified the abnormality, either flow or unselected cells, but whereas in the subsequently in one of the follow-ups, it was a complete relapse. So hence, this is another likely advantage of this application in predicting early response, early relapse. So I would like to summarize what I have spoken so far. As a proof of concept, we established CD19 fish has a diagnostic utility in post-therapy B-cell malignancies. The sensitivity of CD19 fish is high in detecting the in minimal residual disease compared to flow cytometry. The results are comparable with standard flow cytometry, particularly low disease detected by flow. It is very useful. The CD19 selection complements karyotype and routine fish. Potential to recognize clonal evolution in terms of recognizing the subclones has been shown in 11 cases. And also, as an example, we have shown in one case, early relapse can be recognized by using the CD19 fish. Despite the advantages of CD19 fish, there are some downsides one can recognize here. Some of them are outlined here. CD19 fish is limited by probe availability for specific histologic types. Normal range of cutoffs are sometimes a problem in identifying a very low frequency of fish positivity. The CD19 fish is inferior in detecting the abnormalities, the low sensitivity compared to MRD flow. However, you can also state that it cannot completely replace the karyotype because karyotype identifies the genome-wide chromosome abnormalities, whereas fish is limited to a small number of known chromosome targets. In some cases, it's not very useful when the disease is identified by flow cytometry or by unselected samples fish. This may not be very useful. At the end, I would like to acknowledge the people who has participated in this study. From Columbia University Irving Medical Center, Caitlin Walsh and Alicia Cristiano has played a major role in doing all the technical work. Govind Bhagat, Bashir Alabid, Andrew Parrott are the pathologists who has done all the pathology workup and interpretation of the flow data. George Vlad is a flow cytometry person who has done all the flow cytometry. From STEMCELL Technologies, Seema Saw and Benoit Gilbert have helped us in the initial stages of standardization of these procedures in my laboratory, and I obviously thank them for their help. Thank you very much.
Thank you very much, Dr. Van de Valle. We'll now hand the presentation over to Dr. Nurai.
I am going to talk about cytogenetics and enrichment techniques used in myeloma patients in the laboratory system. It is hard not to see the audiences, and also, of course, it's going to be hard not to see me when I'm presenting, but in lieu of COVID-19, this is the best that we can do, so please bear with me. First, my disclosures. The support of this program is provided by Abbott, and I have been requested by Abbott to give this presentation. In this presentation, I'm going to have a brief introduction to myeloma. Next, I'm going to switch to genetics and its role in myeloma. And then a branch in genetics, cytogenetics, its utility, advantages, and limitations. And a sub-branch in cytogenetics, fluorescence in situ hybridization, or FISH, and its relationship to the risk stratification and treatment and management of the myeloma patients. And I'm finally talking about enrichment, its concept and use in myeloma. A brief epidemiology about myeloma: 1.4% of all cancers, around 10% of hematologic malignancies, and but a high mortality rate, 1.9% of all cancer mortality. Lifetime risk, 1 in 143, and in US, based on the 2016 data, there were like over 30,000 of myeloma cases, new cases, which half of them actually died, more common in men than women. And most common in people over 60. Average age of diagnosis is 70, and it is rare under 40 years of age. And it's twice as high in African American population with unknown reason. And unlike the other malignancies, which are metastasized to bone, the osteolytic bone lesions in multiple myeloma have no new bone formation. So, and bone disease is actually the main cause of morbidity in myeloma patients. Multiple myeloma is the malignant neoplasm or terminally differentiated plasma cells, but it's a part of a range of disorders known as monoclonal gammopathy, which actually started with a precursor state of gammopathy of undetermined significance, or MGOS, and then can actually progress to smoldering myeloma and then eventually to multiple myeloma. But MGOS and smoldering myeloma are generally asymptomatic clinically, but not genetically. The 5-year survival rate of people of multiple myeloma is about 50%, which shows high mortality rate for the multiple myeloma. In terms of diagnosis of multiple myeloma, genetics or cytogenetics, they don't have any place. The diagnosis is actually based on clinic, some paraclinic testing, and as you can see at this big, busy table, there are criteria for the diagnosis of the myeloma. However, myeloma genetics is an important piece of the myeloma puzzle. If we could figure out what is the genetic type of myeloma, we can actually help the patients and then predict the pattern of the treatments which can be received by the patient, and also outcomes of the treatment and prognosis of the patients. As you can see in these illustrations, we have primary events in terms of genetics in myeloma, and then we have secondary events. All the primary events mostly are cytogenetics in nature. And then you can actually divide patients into different categories. The top part, it says hyperdiploid ones. And then bottom part, it says non-hyperdiploid, which usually involves with different translocations involve the IGH gene. And all these genetic changes can happen actually at the MGOS level. So as I mentioned, although MGOS and smoldering myeloma are clinically silent, but they are quite busy in terms of genetics. As the disease progresses from MGOS towards smoldering myeloma, you can actually see a new genetic changes, many of them at the molecular level. You could see mutations in NRAS or KRAS. And then when it moves toward myeloma proper, you can see MYC rearrangements can happen, or deletion of the 17p can happen, loss of TP53 gene. And also when it gets to the relapse of myeloma, you can see the host of different genetic changes in the portfolio of the patient. If you look at the top part of these illustrations, it shows the progress of the disease. In the middle, you can actually see the stages of the disease. So at the initiating events, which you have a normal cell which moving toward a new genetic changes, and then the driver mutations and diversification can happen in that cell. Driver mutations are usually those mutations that help the cell survival, cell proliferation, and you can see these are helping the tumor cells. Then tumor can actually, so that's that MGOS level. As you move towards smoldering myeloma, now the bulk of the tumor increases. So the tumoral cells actually increase in population. They do so by reaction with the immune system of the body. As you can see here, the myeloid-derived suppressor cells, the kind of pinkish in color cell, those cells, they have been actually shown that they're increased in number in inflammations and cancers, and it has been shown that an increased number of them can actually worsen the prognosis of the patient. And in myeloma per se, they are actually helping in suppression, tumor suppression, and immune system suppression, and reacting with the T cells. So these are all helping. So you have a driver mutations in order for proliferation and survival of the cells. Now you have this immune suppression in order so let the cells actually grow in number so that the bulk of tumor actually increases. Then when you get to the myeloma proper, you can actually see the changes in clonal dominance with end organ damage. Now we have a myeloma kidney. And these are involvement of the dendritic cells, the kind of purplish color cell there. And so cells are using the immune system of the body in order to increase their population to survive better in the environment and also damage the end organs. And when you get to the high risk state of the disease, like plasma cell leukemia or extramedullary disease or relapse, you can see the host of genetic changes. If you look at the bottom part, like MYC translocations, jumping translocations, homozygous, tumor suppressor gene inactivations, and amplification of 1q. So these are all add up as a secondary events in the genetics of the myeloma. When the physicians dealing with the myeloma patients, what kind of genetic testing actually they order? There's a standard approach, as you can see in the left column, as a desirable test. So they order chromosome analysis, they order FISH analysis, and they order plasma cell specific FISH analysis, which actually involves with that enrichment processes that I will get to it. On the left-hand side, you see other investigational tests for trials or non-clinical settings, but some of them actually used for the clinical settings as well, like gene expression profiling of myeloma patients, or chromosome microarray, or next-gen sequencing. So cytogenetics in myeloma is a big important piece. As you saw in a previous slides, the cytogenetics changes are actually started as a primary event. So myeloma itself is characterized by several cytogenetic abnormalities that occur at various time points of the disease course. The interpretation of cytogenetic results is complicated. The reason why there's a complication there is that there are multiple different cytogenetic abnormalities and the complexity of those abnormalities altogether. The method that you use to detect those abnormalities and also the disease stage at which the patient are detected, those abnormalities are detected in the patient. So altogether, putting all these together make the interpretation a little bit more complicated. Specific cytogenetic abnormalities affect clinical presentation of the patient, progression from smoldering myeloma to multiple myeloma, or prognosis of the myeloma and patient management, as I will talk about these. As I mentioned prior, some abnormalities start at the time of initiation of transformation of the normal plasma cell toward the abnormal plasma cell. And to the level that you have a limited clonal proliferation, this is the MGOS level. These abnormalities, these cytogenetic abnormalities called primary aberrations or abnormalities. But then as the disease progresses, and then later on the course of the disease, there are other abnormalities, other cytogenetic abnormalities add up to this process, and then these are actually called the secondary ones. These cytogenetic abnormalities in myeloma affect every aspect of the disease, from evolution to the malignancy, to clinical presentation, response of the patient to the therapy, and also the outcome and prognosis of the patient in total. As I mentioned, the primary ones classify MGOS and multiple myeloma into several distinct, mostly non-overlapping subtypes. So they are helping to put them into different groups, at least. They are thought to occur at the time of the MGOS and are believed to have a role in the initial pathogenesis of MGOS because they are primary events there. The secondary ones, they can actually occur at, in any of the primary subtypes of the myeloma, and it can influence the outcome of the disease. On the contrary, with the primary cytogenetic abnormalities, they are overlapping, and many different, secondary cytogenetic abnormalities can occur in the same patient. I'm choosing this image from this nice paper in blood cancer. There are terminologies that used in some papers. They are not, I would say, accurate cytogenetics terminology. I'm using the cytogenetic terminology rather than the popular one. Here on the first box under the image, you can see that says trisomies or any of the IGH translocations. Instead of trisomy, I'm using hyperdiploidy. Hyperdiploidy means any counts over 46, which is a normal count of our chromosomes. And then, of course, majority of the hyperdiploid cells in myeloma are actually composed of trisomies, means one extra copy of some of the chromosomes, which usually odd number of the chromosomes. But you could see tetrasomy, pentasomy, and then all could actually fall into the hyperdiploidy. So I'm using a more broader terminology as a hyperdiploidy. So as a primary, as a normal plasma cells can actually occur these abnormalities in like hyperdiploidy or any of the IGH translocations or combination of these two. Then you switch off to the middle box. It says the secondary cytogenetic abnormalities. Here is at the time that MGOS or smoldering myeloma are switching off to myeloma or progress to the myeloma. Here you're going to have the secondary ones like deletion of the 17p or gain of 1q. Again, the terminology for gain of 1q is 1q amplification. Amplification means several copy, a numerous copy of the exact point of any of the chromosomes and then multiple of those copies. But in myeloma, we just gain one or two extra copies of 1q. So better to call it a gain of 1q rather than amplification. And also the translocation 414, which are associated with the high risk disease in myeloma. On the last box, you can see the secondary cytogenetic abnormalities occur with progression. Here you have the relapse or refractory type or plasma cell leukemia. These are more actually of those secondary changes. Some of the rare ones can actually occur at this point. Um, what happens to the cytogenetic abnormalities in those stages? As you can see in the middle of the illustrations, you can see that you have two major subtypes. The red box says 14q32 translocations, means translocations with IGH gene, and the green box says hyperdiploidy, usually trisomies, but sometimes tetrasomy of odd number chromosomes. And also you have a dotted red line there, and that actually shows that there are some overlap of these two. So the patient could have hyperdiploidy as well as some of the translocations. But regardless, these are two actually distinct groups. But this distinct group, they keep the difference during the MGOS and myeloma, but when it gets to the relapse, you can see that there are all other secondary abnormalities that can happen and can add to any of these groups. So, for instance, MEK rearrangements or mutations of RAS or BRAF or deletion of the TP53. So now, regardless which group they are belong to, they still could have these abnormalities. So in this nice table shows the primary cytogenetics classification of multiple myeloma. Based on that, you can actually classify these. On the first row, you see hyperdiploid myeloma, and I explained trisomies or sometimes tetrasomy or pentasomy, but within the box of the hyperdiploidy of odd number chromosomes, that happens in 42% of the patients. 30% of the patients, they have some kind of translocation with IGH, which the breakdown of those, some of them are more common, some of them are less, but generally altogether, like 30%. 15% of the patients, they have a combined of these translocations and hyperdiploid on the background. So altogether, almost 90% of the patients of myeloma actually belong to one of these two groups or an overlap of those two. Another 10%, they could have either a rare type of translocation with IGH or monosomy 14, or be completely normal in terms of FISH or cytogenetics finding, which is like 3% of the patients, or some other abnormalities which are not actually hyperdiploidy or IGH translocations. So as you can see that in majority of the myeloma patients, cytogenetic abnormalities are present and detection of those is necessary. Secondary aberrations, there are numerous of them described in myeloma, and as I explained, one or more can occur in any of the primary cytogenetic subtypes of myeloma, I mean the MGOS or even smoldering myeloma. One of the most common one is monosomy 13 or 13q deletion. Previously, they thought that it has a significant adverse prognostic effect, but then they realized if you detect it by chromosome analysis, then that's the time that it has an adverse prognostic effect, not by FISH. And then reason of that is mostly it's because, as I will explain, plasma cells cannot grow in cytogenetics culture, so and they cannot actually generate proliferating cells, which can actually have some chromosomes to be studied on. So the fact of the matter that in a patient, you could actually have an abnormal karyotype, which shows that you're dealing with a more proliferative type of plasma cells by itself is a poor prognosis. And also, finding of monosomy 13 is also sometimes in a context of hypodiploidy or some of the IGH translocations. So these are all add up to the adverse effect of monosomy 13 or deletion 13q detected by chromosome analysis rather than by FISH. The other secondary abnormality, which is very commonly happen, is deletion of the 17p or short arm of the chromosome 17 with loss of the TP53 gene, which typically occur later in the disease course. But also, if you find a deletion of the 17p in a different stage of the disease, it could mean a different meaning for the patient's prognosis. For instance, detection of the deletion of the 17p in a smoldering myeloma indicates higher risk of progression to myeloma. But if you found it in the myeloma itself at the time of the multiple myeloma, it indicates adverse prognosis of progression-free survival or overall survival. A duplication or gain of the 1q, it's noted in more than 40% of the myeloma patients, but it's interesting that it does not, you cannot find it in MGOS level. So 0% of MGOS level, patients have 1q, the gain of 1q, but you can see it in a smoldering myeloma or multiple myeloma. This fact by itself shows that gain of 1q it has a role in disease progression and associated with a higher risk of progression in smoldering myeloma and also the unfavorable outcome for the multiple myeloma. The other secondary ones, the common ones, are the MEK translocations, so comes with the rearrangements of the MEK gene and also the deletion of the 1p or a short arm of the chromosome 1, that they both have adverse prognosis in multiple myeloma. So in this table, you can see on the left-hand side, the column of the left-hand side shows the different cytogenetics abnormalities. On the two right-hand side columns, you can see the smoldering myeloma versus multiple myeloma. If you look at this table, the stage of the disease that the patient in and the abnormality could have a different meaning. For instance, if you found translocation 614 in either smoldering myeloma or multiple myeloma, prognosis is not bad. But if you found 1416 and a patient is in smoldering myeloma stage, it has a standard risk. But in multiple myeloma, it shows a high risk. You can see the ones in red are basically the high red ones. And then you can actually see the 1420 has the similar effect, a standard risk in a smoldering myeloma, but high risk in multiple myeloma. So it's important. It's one of those complexity of interpretation of the results of cytogenetics. You have to actually know which stage patient in, so you can actually relate this abnormality to the prognosis or risk of progression of the patient in that stage of the disease. And the clinical staging system of the myeloma is also revised, and cytogenetics still has a role in it. So there are different laboratory tests and a clinic involved in determining the staging of the myeloma, but no high-risk cytogenetics versus high-risk cytogenetics findings. It can put the patient in a stage 1 versus 3. In terms of risk stratification, the usual practice is that they put patients in a good, intermediate, and high risk. So anything which no FISH or chromosome analysis abnormalities and hyperdiploidy, translocation 1114 and 614 put the patient in a good risk group. And then high-risk groups are the ones with the deletion of the 17p, 14, 16 translocation, deletion of 13, hypodiploidy, or gain of 1q. And then they put the translocation 414 in an intermediate group. However, Mayo Clinic does not like this. They put them in two risk groups, standard versus high risk. They push the 414 into the high risk group, and then 75% of the patients, they show the standard risk abnormalities, hyperdiploidy, 1114 translocation, and 614, and the 25%, they have all different kinds of translocations and gain of 1q, deletion of 17p. Also, as much as it's introduced in lymphoma as a double or triple hit, you can have multiple of these factors together, so then that's actually pushed the patient to the high risk group. The cytogenetics also has a role in the management of the patient, even in treatment of the patient. As you can see here at the top rank, you have the standard risk abnormalities, intermediate one, and a high risk one. And then as you can see, not totally different, but a patient can be managed in a different way based on the cytogenetics abnormalities that they found in the patient. So that goes with the patient management and treatment. So until now, I showed that how cytogenetics abnormalities, especially detected by FISH, are important in risk stratification, determination of the disease progression or stage of the patient that the patient is in, management of the patient, treatment of the patient, and risk stratification. So when it comes to the testing for clinical purposes, cytogenetics in myeloma can be assessed by either chromosome analysis or fluorescence in situ hybridization or FISH, or combined of these two. However, between these two, the most commonly run is FISH. Reason why is the chromosome analysis needs proliferating cells in the cytogenetics cultures in order to be proliferated, get to the uh, proliferation stage, to the phase of mitosis, so we can actually arrest the cells there, harvest or collect the chromosomes at that level, and do the chromosome analysis on. But plasma cells are highly mature cells, and they are rarely grow in a cytogenetics culture, even if you add the mitogens to them. Because of that, almost 90% of the time, you're going to have a karyogram, normal karyogram, in a patient referred for myeloma. Reason why is that the bone marrow contains other cells that they can actually take over the culture and generate a normal karyogram instead of the plasma cells, which are abnormal. But still, chromosome analysis could be helpful in a way that just the matter of the fact that you have an abnormal karyotype shows the highly proliferative plasma cell as the original neoplastic cell, and then that can actually put the patient in an unfavorable category. Besides, during the treatment and management of the patient, many patients actually develop MDS. And an MDS has defined cytogenetics aberrations, and you can actually detect it by the chromosome analysis. Also, as I mentioned, the prognostic effect of some of the abnormalities that you can actually detect by FISH, if you detect it by chromosome analysis, have more grave prognostic impact of the patient. And if abnormalities of any kind are detected on chromosome analysis in a patient with suspected MGOS or smoldering myeloma, that again shows the high risk of the progression because, like I said, you can actually know that that plasma cell responsible for this disease in this patient is more proliferative and more dangerous and can actually progress to myeloma faster. However, most of the burden on the testing is on FISH. The advantages of the FISH testing is you don't need the proliferating cells. You can do the FISH on the interphase nuclei. You can even do it on the dead cells. Of course, you have to preserve the cells to do the testing. It has higher resolution of the chromosome analysis. Chromosome analysis is taking a look at the whole genome as a far distance. FISH gets a little bit closer. And compared to chromosome analysis, FISH can detect lower level of aberrations. For chromosome analysis, we analyze 20 cells, but for FISH, at least 100 to 200 cells. So you can actually have a higher chance in a random testing to get the abnormal cells present in a lower level. It has its own limitation. FISH has a tunnel vision. So you test one thing and you get the answer for that, not the whole genome. For chromosome analysis, you look at the whole genome. So you can hunt for the abnormality. But for FISH, each test has its own answer. Is there a translocation 1114 this patient? Yes or no. So I do the FISH probe for that and I get the yes or no. But is that patient has a deletion of the 17p2? I don't know. I need to do tests for that one too. So then usually FISH testing is a panel. So you have your multiple probes toward the target abnormalities. And also FISH is very sensitive. If you do the FISH and if your patient gets a few cycles of therapy before FISH testing, that can actually change the genetic composition of the patients of the cells, and then you're gonna lose the valuable data that you can actually get it prior to that. So then critical moment for FISH testing is at the beginning of the disease and also at the time of the relapse. And I put this one in a red sentence as statement as in cases with a very low level of involvement of the marrow, still FISH gonna miss the abnormality because FISH is bound by the cutoff values. You see in any laboratory test, you can see a range for the normal range. But for FISH, that range does not exist on the commercial kits that you buy from the vendors. You have to define your own range of normality for FISH testing, and that's called cutoff value. Each lab has to define that cutoff value. So if the abnormality is lower than the cutoff value, it's not going to be reported out. So very low level can be missed. So for this purpose, is enrichment a solution for this very low level of bone marrow involvement? Bone marrow contains several different types of cells. I like the jelly beans analogy. So you can have multiple colored jelly beans, multiple different cell types. However, in most of the malignancies, only in one cell type with a unique antigenic characteristics that is responsible for that neoplasm. let's say they are black in color. So if the cell of interest exists in a small population, now below the cutoff value of the fish, now you have a bowl full of jelly beans, only one of them is as your target cell. So if you start counting these and if you start searching inside a bowl and then stop after 100 or 200 cells, maybe the abnormal cells is number 201 and you're missed. So for that purpose, I think it's better to actually handpick those cells, take them out of the mix. It's like a bowl of jelly beans. You take the black one out of the bowl, and now you can actually directly test on those cells and detect the abnormalities. Now, any low level, you still have the all abnormal cells in front of you, and then you can see if they are abnormal or not. I mean, no plastic cells, they are abnormal or not. Um, uh, sorry about that. That was my timer. So I need another 5 minutes to finish. Cells in our body carry antigenic markers on their surface, and one of them is the cluster of differentiation CD, or the ones which actually detected by flow cytometry. And then if we have a unique antibody, a monoclonal one, against that CD, we can actually select that cell. And that's actually the way that a system of enrichment actually works. So you have a monoclonal antibody against a specific unique CD for the cell of interest. In our case, plasma cells, CD38. So that monoclonal antibody you add to the mix, and also you add some magnetic bead to the mix. And then now you have this picture at the bottom. You have the cell with the antigens on the top. So the monoclonal antibody attaches to that CD38, but at the other end attaches to the magnetic bead. Now, if you pull the cells through a magnetic field, all these beads will attach to the inner surface of the field, and the rest of the cells are go out. So in this way, you actually handpick your cells of interest, attach them to the wall of the magnetic bead. Now, if you wash that wall, the pellet of the cells that you get, these are all your neoplastic or targeted cell, whichever it is. In this case, all plasma cells. Now, if you perform FISH on those, now I can see the abnormalities. For instance, this is a plasma cell enrichment case, and this is an 1114 translocation probe. As you can see here, there are two fusion signals, one red, one green. That shows there is a translocation 1114. Or any other probes, like 13q deletion here, one red, two green. One of the red is missing, so it's a deletion of the 13q. Or one red, one green is missing. That's the monosomy 3, so 13. And a gain of 1q here with the 3 red signal shows one extra signal for the long arm of the chromosome 1. Or trisomy 5, that's in hyperdiploidy, you can actually see that. Or trisomy or tetrasomy 11q again shows in the hyperdiploid cases. Or translocation 1416 again, these are all can be detected by this technique. How effective is this? You can imagine that you're handpicking the abnormal cells, and then you do FISH technique directly on those cells, and now you look at it under the scope. So you're not losing anything, any cell under cutoff value, low level abnormalities, you still can see it. And I'm going to share you with the study, a retrospective study that we did in my previous company that I worked for. Almost 3,000 patients, half of them prior that establishment of enrichment in our lab, and then half after establishment of enrichment in our laboratory. And we compare how many actually, how many percent of the cases had abnormality by FISH without enrichment or with enrichment. As you can see on the left-hand side, we have myeloma cases referred to our laboratory prior to establishment of the enrichment. So we didn't enrich those cells and directly FISH on those cells, and almost close to 30% of them showed abnormalities. Do you remember that in cytogenetic abnormalities is seen in almost 90% of the patients? So here we couldn't actually detect them because they are very low level. At the right-hand side, you can see not the same patients, but a group of the patients, 1,500 of them, that we actually did enrichment and then did FISH testing on the enriched cells, and that's over 80% shows the abnormalities. I mean, the difference is humongous, and you can see how effective is the enrichment to address the low-level abnormalities caught by FISH. And this concludes my presentation. Thank you for listening, and goodbye, everyone.
I would just like to thank everyone again for joining us for our workshop today.